when I first started giving these talks, I said, if you have type 1 diabetes, don't ever even think about carb restriction or the ketogenic diet. So I've changed my tune about that. There are now a publication authored by David Ludwig from Harvard to show that carbohydrate restricted diets may be optimal for those even with type 1 diabetes. So a little bit about ketones in general. Ketones, we know, are energy substrates, and now more recently in the last 4 or 5 years, we come to understand them as a powerful signaling molecule that functions almost like a hormone. There's a ketone receptor. There's several ketone receptors. Ketones function as histone deacetylase inhibitors. Ketones function to suppress inflammatory pathways that in a way that's completely independent of their metabolic function. So they have important signaling properties. Clinical ketosis is defined as a blood level of ketones that's above 0.5 millimolar. So that takes about, if you stopped eating now, if you're not on a ketogenic diet regularly, if you stopped eating now, it takes about 24 hours for your blood ketones to really get up into the clinical ketosis range. So you could achieve it with fasting, and if you're low carb, you would get into ketosis faster if you're doing intermittent fasting. Some people do urine ketones. That's okay. It's not optimal, but a urine ketone level of 15 milligrams per deciliter on the keto stick that you can buy at any CVS or Walgreens will correlate to about a 0.5 millimolar ketone level. Nutritional ketosis, I define it as a dietary strategy to elevate ketones into this range, and that typically involves a carbohydrate consumption of fibrous carbohydrates, non-starch, non-sugar, of less than 10% of the total diet, and generally speaking, 25 to 50 grams of carbohydrates. That is the level where you start to get into nutritional ketosis and start to get some of the benefits of nutritional ketosis. Ketoacidosis, and this comes up a lot with the medical students, or if I teach at a clinical conference, all the hands go up, well, what about ketoacidosis? Ketoacidosis is a very specific metabolic derangement associated primarily with the absence or insufficient amount of insulin in the body. So without insulin, when our ketones level rise, that actually releases a small amount of insulin, which helps your body fine-tune ketogenesis in the liver. So without insulin, we have runaway ketogenesis. And in the context of high glucose, high ketones can be very detrimental to health and create an acidotic situation that, if it's not corrected with fluid, electrolytes, and insulin injection, will lead to coma and death. So so even, I was going to say, even people with type 1 diabetes can successfully do the ketogenic diet if they manage their insulin correctly. Keto adaptation is a time-dependent physiological adaptation to using fatty acids and ketones for energy. So we know that the more you do the ketogenic diet or carbohydrate restriction, the more you adapt, the easier it gets, and the more benefits you derive from those adaptations. And the last thing I want to highlight is exogenous ketones, because this term did really not exist years ago, like 10 years ago, and now they're on the market. I get a lot of questions about it, and there are a lot of research being done. I think there's at least a half dozen registered clinical trials on clinicaltrials.gov right now looking at exogenous ketones. So there are different strategies that one can use to get into a state of nutritional therapeutic ketosis. There is starvation, that's not fun, but there's caloric restriction, there's intermittent fasting, there's the ketogenic diet. So now we know, I'm just coming from an epilepsy conference, that the modified ketogenic diet, which is what you see here, or at least the ratio is about 75% fat, 20% protein, and a minimal amount of carbohydrates, like 5%, seems to have similar seizure protection in adults and maybe in some pediatric populations too, depending on the disorder. So these things, basically starvation and the ketogenic diet and intermittent fasting, mobilize fats. The fats go to the liver, and it stimulates beta oxidation of fats with the continuous suppression of insulin. So only in the context of mildly depleted liver glycogen and continual suppression of the hormone insulin, and blood glucose drops a little bit too, but the homeostatic mechanisms that maintain blood glucose are very powerful. So blood glucose really does not bottom out. It might stay in the 70 to 80 milligram per deciliter range, even with no carbohydrate consumption for many people. So these ketone bodies are produced, beta hydroxybutyrate, acetoacetate. Acetoacetate can spontaneously decarboxylate to acetone. Interestingly, acetone has some interesting functions on various ion channels, which can affect brain function. And this is difficult for patients to achieve. So we developed various forms of exogenous ketones, and also acknowledged that fats, ketogenic fats like medium chain triglycerides, which go directly to the liver via hepatic portal circulation and are oxidized very fast to ketone bodies. They're not packaged into chylomicrons like the long chain fats. And then ketone salts and ketone esters are metabolized partially in the liver, but mostly spill into the blood and then rapidly elevate ketone levels. Like a ketone ester If a person consumed it right now on stage, by the time I get off stage, it'll look like I fasted for 10 days. So you can achieve a level of ketosis in the 5 to 6 millimolar range, go from 0 to 5 to 6 within 30 minutes. So they are very powerful agents that we're studying. But as the science sort of advanced, we understand these molecules not only as energetic substrates for the brain, the heart, and even skeletal muscle, but they have powerful signaling properties in the brain and in other tissues. So I'm not going to go through all the signaling pathways here, but you can see in this panel below that there are many different signaling pathways that are impacted by nutritional ketosis. So I've given talks at pharmaceutical companies where they want me to give a talk on the mechanism of the ketogenic diet so they can sort of reverse engineer some drug to target a mechanism. And when I throw up, that panel there that shows all the mechanisms, I think they get depressed because they realize a drug's really not possible. So here's the biogenic or bioenergetic effects of ketones. And in this case, it's just the brain. So the liver is the site of ketone production. So under situations of suppressed gluconeogenesis in the liver and also suppressed insulin signaling and mild glycogen depletion, the liver produces these ketone bodies as acetyl-CoA from beta oxidation of fatty acids accumulates. And the acetyl-CoA condenses together to form acetoacetate and then beta-hydroxybutyrate. Beta-hydroxybutyrate is the primary ketone body in circulation because it's more resistant to degradation, whereas acetoacetate can be spontaneously decarboxylated. But beta-hydroxybutyrate needs to be broken down to acetoacetate to be used as fuel. So keep that in mind. But acetoacetate is essentially the oxidized form of beta-hydroxybutyrate. So the point is the liver makes ketones but does not use ketones as an energy source. It lacks succinyl-CoA transferase, and that's very interesting because cancer cells also lack this enzyme. Most cancer cells lack this enzyme that allows the cancer cells to use the ketone bodies for fuel. So these ketone bodies spill into circulation and they bypass many of the regulatory steps associated with the glucose being able to support bioenergetic functions. So in the case of impaired GLUT1 glucose transporter 1 deficiency or GLUT3 deficiency, that's the transporter on the membrane of neurons. In the case of those being deficient or not functioning, or in the case of Alzheimer's disease or traumatic brain injury, the GLUT3 transporter gets internalized inside the cell, and then the glucose can't get into the cell. But ketones bypass that process. It even bypasses pyruvate dehydrogenase complex. So if you have pyruvate dehydrogenase complex deficiency syndrome is treated with the ketogenic diet. So for kids that have this, they need to be on the ketogenic diet. PDH is also impaired in Alzheimer's disease and also traumatic brain injury. So, and Alzheimer's disease. So we know Alzheimer's disease is characterized as glucose hypometabolism. And the hallmark characteristic of Alzheimer's disease is a decrease in intensity of a fluorodeoxyglucose PET scan. So there's less of an intensity showing radioactive glucose present in the liver. There's less uptake of glucose. It's also interesting to observe that a number of studies showing that cerebral blood flow increases about 30% during acute hyperketonemia. I looked this week, there's over 30 clinical trials on using the ketogenic diet in cancer studies. So this is a very emerging field, and I think you're going to see with the clinical trials now a lot more studies, results from those studies hitting PubMed. So it was observations that we made in the cell types that we studied under hyperbaric oxygen therapy and also with supplemental ketones. We observed that ketones decrease proliferation in these cancer cell types. And the only thing that really explained the observations that we saw, especially the damaged mitochondria and the overproduction of oxygen-free radicals as we increased oxygen concentration. I have published at least 7 articles or studies with Tom Seifert, and we work together in different capacities. But he explained to me the Warburg effect, that cancer mitochondria or cancer metabolism is fundamentally different from metabolism of healthy cells. So essentially the Warburg effect in one sentence is damaged mitochondrial respiration and there's compensatory fermentation. So the basic energy processes that allow a cell to maintain its bioenergetic potential would be oxidative phosphorylation. The mitochondria is making about 88 to 90% of the ATP, the energy currency in the cell. As a person or the cells are exposed to a number of different agents, they could be chemicals, radiation, inflammation, hypoxia, insulin resistance, and hyperglycemia produces ripe environment for the mitochondria to be damaged and the mitochondria DNA to be damaged. The nucleus has very robust DNA repair mechanisms. The mitochondria does not have as robust DNA repair mechanisms. So if a cell is bombarded with things like radiation or carcinogenic agents, the capacity for the mitochondria to repair itself is not as high, is not as robust as the nucleus ability to repair DNA. So the mitochondria take a big hit, and as mitochondrial function is impaired by progressive damage to environmental agents, viruses, for example, can cause cancer, and the viruses that cause cancer impair mitochondrial function. So mitochondrial function goes down, cellular ATP levels go down, and the nucleus of the cell essentially can sense the bioenergetic potential of the cell, it can sense the ATP levels, and it senses that the cell's in an energetic crisis. And when it gets to this threshold where progressive damage to mitochondrial function causes a cascade of events to activate a number of genomic pathways that stimulate the cell to increase glycolysis. And various oncogenes are associated with increased glucose metabolism. So a normal cell then transforms, and understanding too that embryonic cells that are proliferating and growing fast also have a glycolytic phenotype. But normal cells that are not proliferating primarily get their energy from mitochondrial oxidative phosphorylation. When the mitochondria are damaged by a number of different agents, I believe that hyperglycemia, hyperinsulinemia, mitochondrial syndrome is a major driver for this mitochondrial damage. But when a normal cell, when it's activated, it becomes a tumor cell. And there are a number of factors that can drive the Warburg effect and actually make that tumor an expanding biomass to a large solid tumor. And drivers of the Warburg effect can kick on invasiveness and metastasis, where those tumor cells get in circulation and then metastasize, and then it becomes sort of an irreversible process. The metabolic theory of cancer sort of posits that it's the initial damage to the mitochondria that's the enabling factor that essentially transitions a normal cell to a cancer cell. There are genes involved, no doubt, but the metabolic control of those genes is likely sort of the root cause. The geneticist, in years past, it was really just linked to genetic alterations, but now we have an appreciation and an understanding and NIH-directed research to understand how metabolism is directing those gene pathways to actually initiate carcinogenesis and the factors associated with cancer progression too. So damaged mitochondria, I believe in the theory of metabolic theory of cancer, is the initial cause and also a major driver. There's a derangement of tumor metabolism. Tumor hypoxia. As a tumor expands, as the biomass expands, the core of that tumor becomes hypoxic, damages the mitochondria more, there's more genetic mutations, and that the inside of the tumor takes on a more aggressive Warburg phenotype. So it's literally fermenting sugar, as it grows. And really people with advanced tumors, if they look at the actual tumor, the mitochondria are deficient, are structurally and biochemically abnormal. When it comes to aggressive tumors, they have never found a tumor that has what we would call normal mitochondria. So really, the damaged mitochondria are really a major driver of cancer. If your mitochondria are healthy, they call the shots. Healthy mitochondria will keep a high bioenergetic state of the cell. high ATP levels will enhance the fidelity of the nuclear genome such that DNA repair processes will happen and preserve that genomic stability. So that's a really important point, I think, that Tom Seifert tries to make, is that the ultimate tumor suppressors are healthy mitochondria. And there are different ways: exercise, CrossFit, ketogenic diet, low-carb nutrition, intermittent fasting, all these things we know, periodic caloric restriction, enhance mitochondrial function. Things like elevated insulin, glucose, lactate, increased PI3-AKT-mTOR pathway is a major driver for cancer. There are drugs being developed that target this pathway by Lou Cantley, for example, one of our collaborators. And these drugs, interestingly, do not work in the context of a normal diet. They need to be used in the context of a diet that suppresses insulin signaling. So the ketogenic diet dramatically enhances the effect of these metabolically targeted drugs, the PI3 kinase inhibitors. Elevated ROS and inflammation, so reactive oxygen species overproduction kicks on inflammatory pathways, which can damage the mitochondria and really stimulate this carcinogenesis. Suppressed anti and tumor immunity. So as the tumor pumps out lactate and lowers the pH, that actually changes the microenvironment to prevent your body from recognizing that you have a tumor. And the ketogenic diet increases cancer-associated immunity. So it helps increase the vigilance of your immune system to recognize cancer and to attack it through a number of mechanisms. And my colleague Adrian Scheck, formerly at Bioenergetic Institute, had studied that and published on that. So in getting back to how we can change this idea of changing metabolic physiology to not only change brain energy metabolism, but to target the Warburg effect. But keep that in mind as I go through the things that are associated with the ketogenic diet. So we know that about when we give exogenous ketones, we can lower blood glucose by 50%. There was also a paper, an exercise science study, showing that the ketone ester, beta-hydroxybutyrate monoester, can cause a 50% reduction in lactate. We know that glucose and lactate are major drivers for cancer growth and proliferation. We know that there's a reduced proliferation and glycolytic ATP levels in the presence of ketones. Actually, Dr. Feynman here published an article showing that acetoacetate, one of the ketone bodies, could reduce proliferation and reduce glycolytic ATP production. And there's a reduction in glutamine, c-Myc, and various pathways associated with cancer cachexia. So ketones directly affect a number of pathways associated with muscle wasting. And that's the project of my PhD student, Andrew Kutnick, is really studying sarcopenia and cancer cachexia. So, and this is a diet. So this is a diet, and this is administration of a metabolite that your body makes, is really changing these pathways. There's a reduction in proliferation and metastasis, and a reduction in a number of different inflammatory pathways and drivers. Enhanced, as I mentioned, my colleague Adrian Scheck looked at enhanced tumor-associated immunity. And the ketogenic diet caused a dramatic increase in the body's ability to recognize cancer. And this is in animal models, that particular study, but they're studying it in humans now. And it also has an anti-angiogenic and a pro-apoptotic effect. So that's the ketogenic diet, especially in the context of caloric restriction or periodic caloric restriction, kicks on pathways, again, that associated with the body's ability to recognize and attack cancer. And some of the more interesting things that have emerged in the last 5 years is the effect of ketone bodies working through epigenetic mechanisms. So there's epigenetic enhancement of cellular defenses that are associated with preserving healthy mitochondria and perhaps extending longevity or health span over time. And there was a study that I was involved in. It was actually published in Nature Medicine, looking at the effect of beta-hydroxybutyrate on suppressing an inflammatory pathway associated with driving cancer growth. And that's the NLRP3 inflammasome. So when that protein complex gets activated, it kicks on and elevates many different inflammatory cytokines, including things like IL-6 and IL-1 beta. So ketone body specifically, without metabolic regulation, specifically regulates and suppresses. that pathway. So now there's a much greater appreciation for the types of fats and how these diets are formulated. It's also acknowledged that the ketogenic diet does not have to be as strict, 90% fat, as we once thought. So modified forms of the ketogenic diet, actually a form that I follow, which is about 75% fat, is being used clinically now. And there's a number of drugs that I mentioned before mimic many aspects of nutritional ketosis, and that would be 2-deoxyglucose, perhaps 3-bromopyruvate. The jury's kind of still out on the utility of that. Drugs like lonidamine that inhibit hexokinase 2. Metformin has a mild calorie restriction sort of effect, metabolic effect, and we're studying that too, and we've published some research on that. So sort of the gist of this review, this publication, was that the ketogenic diet could target the Warburg effect, and then that it could be a very powerful, at least an adjuvant to the current standard of care. The message that we wanted to convey in this review, which was, co-authored with just conventional neuro-oncologists and people who really didn't even study the ketogenic diet, but our research and some of the studies that have been out there convinced them that this was a very viable approach, especially for brain tumor patients that have epilepsy. They have seizures too. So they have two things that the ketogenic diet can work very effective for. They have seizures caused by the brain tumor and a rapidly expanding, highly glycolytic tumor. So it just makes sense that brain tumor patients, not all cancers are as responsive to ketogenic diet therapies. There are cancers like liquid cancers like leukemia, lymphoma, testicular cancer. These things are very responsive to the standard of care. Not that they shouldn't use the ketogenic diet, but they should consider the standard of care, the frontline approach. The slide that I showed you previously really went through all the different mechanisms and clearly demonstrates by published literature, some of it is animal model studies, some of it's in human, that nutritional ketosis or therapeutic ketosis targets all the hallmarks of cancer. So when a drug company is developing a drug to target cancer, they're usually targeting, invasiveness or angiogenesis. The ketogenic diet, especially if it's calorie restricted, targets all these things. And this is research that my student who published in the Cell Journal, a review on the anti-catabolic effects of ketone bodies. Andrew Kutnick, who's type 1 diabetic, uses nutritional ketosis to manage his type 1 diabetes. So ketone bodies, we know, decrease nitrogen excretion. It reduces alanine mobilization. So the major gluconeogenic amino acid would be alanine. So alanine goes down quite remarkably if you infuse ketones. And that was a human study where they IV infused ketones and showed alanine goes down significantly. And one of the most interesting effects of exogenous ketones is that it causes a dramatic effect at lowering your blood glucose levels. So in animal models, in rats, for example, that have a blood glucose of 120 to 150, if you give ketone esters to their maximum tolerable dose, the blood glucose goes down to 30 or 40 milligrams per deciliter. That's not changing their diet. That's just a ketone-induced lowering of blood glucose. So this has not been published in humans yet, this dose level. I mean, I've studied it, I've consumed it myself and can show and can demonstrate that, I can get glucose levels down to the range where it just doesn't even read on the meter with certain ketone esters. So I think that's an important therapeutic application. And I know people are studying this now, looking at insulin resistance and diabetes, but it stimulates muscle protein synthesis and increases muscle regeneration and proliferation. We know acetoacetate does. The ketogenic diet has been reported to have anti-Cachexic effects, reducing pancreatic, colon, lung cancer, and skeletal muscle catabolism in rodent models. So we're interested in partnering with the Moffitt Cancer Center to get a clinical trial in humans. Putting patients on the ketogenic diet can be difficult, so they're very interested in supplementation. I'm interested in both, the ketogenic diet and then supplements essentially as an adjuvant to the ketogenic diet.